Let's discuss part D, that will be having 4 more questions. Please refer previous videos for part A, B and C, which will carry 1, 2 and mark 3 marks respectively. So here, this is a question regarding lac operon. I am not going deep into the description. Instead, we'll be jumping into the each and every questions. So see, much more details won't be there in that particular paragraph. Instead, if you know the whole concept properly, you can even don't have to read it very deep. You have to read it, but no need to give your whole time for that particular paragraph. Instead, you can directly jump into the questions. Why is lac operon said to be transcriptionally regulated system? Yeah, it is transcriptionally regulated because if there is a if in this case, I will show you the answer. The presence of lactose and the absence of lactose is actually telling whether it should be transcribed or not. If presence of lactose is there, the scene is different. The absence of lactose is there, then the whole process is different. So instead, I'm telling here the answer. I will club this diagrammatic representation answer for D and A together. So we will discuss it together and we will jump into the answer for B and C. So this is said that the lac operon has to be operationally at very low level in bacterial cell at all time justified. This is something related to permease. Permease is actually permitting lactose to enter into the cell. So a little bit of permease should be there. So if little bit of permease should be there, then lactose should be active. So the question is related to permease. Simply it is related to permease. Permease should be always there to permit the lactose to enter into the cell. So if permease should present, then lac operon should be active every time. This is the answer. So if you have any lack of clarity in this answer, once we discuss this thing, the diagram with the A part, you will get it clarified. Why is the regulator gene lac operon is marked as I? It is related to inhibitor. This is the one from the regulator, inhibitor or repressor is coming and will go to the operator and bind to it and do the process. So let's jump into this diagram. See, in absence of inducer, the part D is regarding diagram. So I'm directly jumping into the diagram and explaining about it. See, please draw and learn the diagram. Sometimes you may read it and go for the exam, but the thing is that this PIPO ZYA, in that way you learn, or else you may know this diagram, but what is to be written inside that you may club. Most of the students do this. That's why I'm telling draw every diagram and learn. So here in absence of inducer, or inducer is here lactose. Lactose is not there. Lactose is not there in the sense that there is no need to produce any kind of enzymes from ZYA. This ZYA are the enzymes which will cleave, especially the from the one that is beta galactosidase, that is Z. This thing will cleave, that will be cleaving the lactose and produce energy for home bacteria. So if lactose is not there, that means absence of inducer, they don't have to be produced. So somehow this transcription process should be shut down. So this you can remember by a small analogy. There is a room with the three electric equipments, that is fan, light and air conditioner AC. So if it is there, if no one is there inside the room, it should be shut down. Okay. If it is someone is entering in presence of inducer or someone is entering, lactose is coming, these three should be produced. At that time, switch on should happen. Here, switch off. Who is doing it? Here it is a repressor. Imagine that in that room there is a person who saves electricity, who love to save electricity, a guard who will switch off if no one is there. And if lactose is coming, lactose will tell the guard to be silent. I am here. I will do whatever I want. I will switch on the lights because I need it. and the repressor will be silent because it's for a purpose. Some purpose is there, electricity is not wasted. So remember to this particular analogy and learn this one. So this guard or repressor will bind if there is no lactose. If lactose is there inside, repressor will be binded with a little amount of lactose and repressor will be silent, so there is no one to block this process. So RNA polymerase will produce what enzymes from ZYA, that is beta galactosidase from Z, permease from Y, transacetylase from A. Transacetylase function is not that much clear in our textbook and all. We didn't explore it. But beta galactosidase is for cleaving the lactose and produce energy. Permease. Permease, as we have discussed, is should be produced continuously, whether a lactose is there or not, because it is its duty is to enter the lactose inside. Hope you are clear with the image or the analogy or the lactose operon. Okay, let's move to another question. There is a diagram. See, here it is equator, and here we have the pole. Equator is mentioned as A, pole is mentioned as B. So from equator to pole, when we move, what will happen? What about the species diversity? Whether it is increase or decrease? It will decrease. We have to tell two reasons for it. The reason is, in equator, less annual temperature is there. Temperature is less there. There is no availability of sunlight. These two things are enough. Okay, right, two reasons: sunlight, lack of sunlight, seasonal changes more, uh, and that availability temperature is very hot. That is not okay for every species to thrive. Okay, the approximate number of bird species recorded in India is about 1,200 species. Okay, and let's move to the or question. Name the region in which the world record shows the greatest biodiversity. That is Amazon rainforest for sure. From the childhood itself, for quiz competitions and all, we are learning this, the diversity of Amazon rainforest. Okay. Thank you for listening. We have completed part D as well. We will move to part E in next video.